The topic that I am going to discuss is all about cell as the basic unit of life. So the properties of life as exhibited by cells can be described by a collection of properties. Cells are very complex and their substructure is highly organized and predictable. The information to build a cell is encoded in its genes. Cells reproduce by cell division. Their activities are fueled by chemical energy. They carry out enzymatically controlled chemical reactions. They engage in numerous mechanical activities. They respond to stimuli, and they are capable of a remarkable level of self-regulation. As to the functions of the cell, it is said to be the basic unit of living organisms, and it always possesses a genetic program which is located inside the nucleus in the form of the DNA or the deoxyribonucleic acid. Cells are capable of producing more of themselves because of these DNA. Cells also acquired and utilize energy. Plant cells acquired energy from the sun, and animals utilizes energy from eating foods. Cells are also capable of metabolism and able to respond to stimuli, particularly from the outside world. As to the cell structure, collectively, we have organelles, and these are specialized structures in cells that perform a specific functions. And usually, these organelles are membrane-bound, such as the nucleus, the mitochondria, and the ribosomes of the cells. Cells are suspended in the cytoplasm, which is a jelly-like substance that holds these organelles. This illustration depicts the different structures of the cell. These organelles here inside the cell are capable of different functions that are very important or vital for the cell function. Another remarkable structure of the cell is the cell membrane. And the cell membrane is the outermost component of a cell. It functions as selective barrier. That means it does not allow all materials to come in and out of the cell. Another function is that it encloses the cytoplasm, where organelles are suspended in it. In cell biology, extracellular means materials outside the cell, and intracellular meaning materials inside the cell. The cell membrane is somewhat called as fluid mosaic model, and it is made up of phospholipids and proteins. Of course, phospholipids gives integrity to the cell membrane, and the proteins functions as channel proteins that allows the transport of larger molecules in and out of the cell. Phospholipids form a double layer or said to be bilayer. Phospholipids contains two regions, the polar and the non-polar. The polar region or polar heads are said to be hydrophilic, that means loving water, and usually it is exposed to water. And another is the non-polar regions, which is the non-polar tails, is said to be hydrophobic or water-fearing, away usually from water, and usually situated inside of the micelles. This illustration is a typical structure of a phospholipid. The polar head, which is hydrophilic, and it contains phosphate containing region, and this portion is the non-polar or the hydrophobic region that is made up of fatty acids. As what I've said earlier, our cell membrane is phospholipid bilayer. That means 2 layers of phospholipids. Other structures that can be observed in our cell membrane, such as the membrane channel, which is a protein that allows larger molecules to move from outside to inside and inside to outside of the cell. Another is that we have a receptor molecule here, which is a glycoprotein that is made up of a complex of carbohydrates and a proteins. This function mainly for cell-to-cell interactions. Another important composition of our cell membrane is the cholesterol. Cholesterol molecules provides additional strength for the cell membrane. In cell membrane, the cell membrane is selectively determines what can pass in and out of the cell. The enzymes, glycogen, and potassium are found in higher concentrations inside the cell, while sodium, calcium, and chloride are found in higher concentrations outside the cell. There are different ways molecules pass through cell membrane. One is directly through diffusion, and water, oxygen, carbon dioxide, and other small molecules can directly diffuse from extracellular to intracellular of the cell. Larger molecules cannot pass through the cell membrane through diffusion, but of course through membrane channels. Membrane channels are proteins that extend from outside of cell membrane to another side. The size, the shape, and the charge always determine what can go through the membrane channels. Example, sodium passes through the sodium channels. Observe in this illustration that the red here are non-lipid soluble molecules that usually diffuse through cell membrane channels. Lipid soluble molecules, these are yellows, diffuse directly through the cell membrane. And it always depends on the concentration gradient of the cell. Another way is through carrier molecules. Carrier molecules usually bind to molecules, transport them across, and drop them off. One example that molecules that uses carrier molecules are the glucose. Also, through vesicles, this can transport a variety of materials, usually fused with the cell membrane. This illustration is an example of a sodium-potassium pump that use a channel for them to transport in and out of the cell. The sodium-potassium pump is very important in action potentials, particularly in the muscle contraction. This illustration is a use of vesicle in transporting the materials in and out of the cell. Notice in this number 1, it's a secretory vesicle that moves towards the cell membrane. So this color blue here is the cell membrane, and this one is the secretory vesicle that has a content. So secretory, that means it secretes outside of the cell membrane. So the secretory vesicle membrane fuses with the cell membrane and later on release the contents into the extracellular fluid. Through diffusion, there are transports of molecule or smaller molecules and lipid-soluble molecules using the process of diffusion. So diffusion is a movement of molecules from areas of high to low concentration. So before we understand these things, we need to have a background, particularly with these words: solution, solute, and solvent. When we say solution, is a solid or a liquid or gas that contains one or more solutes. Solute are substances added to solvent that dissolves. And a solvent is a substance such as water that solute is being added to. Example is that when we add salt or the NaCl to water. So water is the solvent and the salt is the solute. And the mixture is said to be the solution. I guess this concept are very basic enough for us to understand. This experiment will discuss further the concept of diffusion. So just for example, we have a container here with distilled water, and when we drop a salt crystal inside the container with water, a concentration gradient exists between the salt from the salt crystals and the water that surrounds it. In a few minutes, the salt ions, colored green, move down their concentration gradient into the water. And later on, salt ions and water molecules are distributed evenly throughout the solution, even though the salt ions and water molecules continue to move randomly. An equilibrium exists and no net movement occurs because no concentration gradient exists. When we say concentration gradient, this measures the concentration difference at 2 points. So the greater the distance, the faster the solute will travel. And we have the filtration here, which is the movement of fluid through a partition with holes. Another are the types of mediated transport mechanisms. One is the facilitated diffusion. So it is a diffusion with aid of a carrier molecule and usually requires no energy in the form of ATP. Another, we have the active transport that moves substances from low concentration to higher concentration and usually requires ATP. And one example is that your sodium-potassium pump. This illustration is an example of a facilitated diffusion wherein it used a transport channel or a protein channel for the glucose molecule to travel from extracellular fluid to intracellular fluid. So the carrier molecule binds usually in the molecule here, the violet, such as glucose on the outside of the cell membrane, and later on, due to the concentration gradient, which is from higher concentration to lower concentration, the carrier molecule changes its shape and release the molecule on the inside of the cell membrane. And these glucose molecules will later on be used up by our cell for the production of energy in a form of adenosine triphosphate. Another illustration that provides us an example of a type of mediated transport mechanism. Particularly, it is an active transport. This is a sodium-potassium pump. So, usually, 3 sodium ions here, the color pink or color violet, and adenosine triphosphate will bind to the sodium-potassium pump. And the ATP breaks down to adenosine diphosphate, this one, and a phosphate releases energy. That energy will be used to power the shape change in the sodium-potassium pump. After changing the shape of the sodium-potassium pump, the sodium-potassium pump usually allow the movement of potassium or 2 potassium ions binds in its surface, and the sodium-potassium pump changes shape, transporting potassium across the membrane into the cytoplasm. The sodium-potassium pump can again bind to another sodium and ATP, and later on will repeat its activity. Another transport mechanism of molecules through cell membrane is osmosis. Osmosis is actually a diffusion of water across a cell membrane, and it drives by an osmotic pressure. And this osmotic pressure is a force required to prevent movement of water across the cell membrane. Just like in this laboratory experiment. This laboratory experiment is a basic experiment for science majors, students, especially bio students. So here we have the container or the beaker with distilled water and a thistle tube with 3% salt solution. And we cover the mouth of the thistle tube with a selectively permeable membrane. In our case, during our undergraduate years, we use chorizo casing. Another step here, the next step is by putting or by immersing this thistle tube to our container with distilled water, we observe that the salt solution is rising and the water in the beaker is obviously decreasing, causing the concentration of the salt inside the thistle tube decreases. And we observe that water moves by osmosis into the tube until the weight of the column of water in the tube prevents further movement of the water into the tube. The hydrostatic pressure that prevents the net movement of water into the tube is already equal to the osmotic pressure of the solution in the tube. Now let's move on with the types of osmotic solutions. We have three types of osmotic solutions, namely hypotonic solution, hypertonic solution, and isotonic solution. When we say hypotonic solution, it is a solution with lower concentration of solute and higher concentration of water. And usually, if we place a cell into the solution, the water will tend to move into the cell and will cause lysis or bursting of the cell. Another is hypertonic solution, wherein it is composed of higher concentration of solute and lower concentration of water. Again, if we place a cell in the solution, the water will tend to move out of the cell, causing crenation or shrinkage of the cell. Lastly, we have isotonic solution. The isotonic solution has equal concentration of solute and solvent. Thus, water doesn't move if we place cell in our solution. That is why the cell will remain intact. This illustration will help us to explain the effects of the hypotonic, isotonic, and hypertonic solutions on red blood cells. Notice that we have three solutions here. These are hypotonic, isotonic, and your hypertonic solution. In each beaker here, we place a red blood cell. In hypotonic solution, when a red blood cell is placed, that with one having a low solute concentration, water will enter the cell by osmosis, causing, of course, the cell to swell or even burst later on. In a hypertonic solution, when we add red blood cell, the one having a high solute concentration, water moves by osmosis out of the cell and into the solution, resulting in shrinkage of the cell. But in isotonic solution, when we place red blood cell, when we say isotonic, one having a concentration of solutes equal to that inside the cell. So water will moves into and out of the cell at the same rate, so no net water movement occurs and the cell will still be intact and remains normal in shape. Another transport mechanism through the cell membrane is this endocytosis. Endocytosis is a process that brings material into cell using vesicles or transport vesicles. We have two types of endocytosis. We have phagocytosis and pinocytosis. When we say phagocytosis, it is said to be cell eating or solid particles, while pinocytosis is said to be cell drinking or with liquid particles. In reverse, of course, we have exocytosis, and it's a process by which materials out of the cell using a transport vesicle. So this illustration is an example of exocytosis. This one is an example of endocytosis. After we discuss the membrane that surrounds the entire cell and the cytoplasm containing different organelles, we will tackle these organelles one by one. Starting, of course, the cytoplasm. The cytoplasm, it is located inside the cell, and it is a jelly-like fluid that functions to give the cell shape and hold the different organelles in place. Another is the nucleus, which is located in the center of the cell, and all cells contains nucleus at some point, particularly eukaryotic cells with true nucleus. The nucleus houses the genetic material, which is the DNA. Letter A in this illustration is the nuclear envelope that consists of inner and outer membranes which become fused to the nuclear pores of the nucleus. The nucleolus, this one, the red one, is a condensed region of the nucleus not bounded by a membrane and consisting mostly of ribonucleic acid and proteins. This one is a transmission electron micrograph of the nucleus, and another is a scanning electron micrograph showing the membrane of the nuclear envelope and the nuclear pores. The nuclear envelope, of course, as what I've said, it is located in the edge of the nucleus. It covers the entire nucleus of the cell. The nuclear pores is located in the surface of the nucleus, and it functions where materials pass in and out of the nucleus, particularly the movement of mRNA from the nucleus going to the cytoplasm of the cell for further DNA transcription. These are the structures that are present inside our nucleus. Number one is the chromosome. And the chromosome is a condensed DNA and proteins that are usually visible during cell division. And the function of the chromosome is that it takes part in the genetic makeup of every organism. Chromatin, on the other hand, is also located inside the nucleus, and these are loosely coiled chromosomes. So this illustration gives us an idea of the organization of the genetic material located in the nucleus. From a fragments of genes that made up our DNA, it will be coiled into a protein known as the histones and will coiled further and tightly packed to form the chromosome. And as what I've said earlier, this chromosome can be visible under the microscope if the cell will undergo a cell division. Another structure inside the nucleus is the nucleolus. And the function of nucleolus is to produce ribosomes, which is responsible for the production of proteins. And ribosomes are attached usually to the rough endoplasmic reticulum and or located in the cytoplasm of the cell. And again, it produces proteins. This illustration gives us an idea of the process of the production of mRNA. So here, the ribosomal proteins from cytoplasm produced actually in our cytoplasm are transported through the nuclear pore of the nucleus and going to the nucleolus of the nucleus. So the RNA or the rRNA or the ribosomal RNA, most of which is produced in the nucleolus, is assembled with ribosomal proteins to form a small and large ribosomal subunits. The small and the large ribosomal subunits will leave the nucleolus and the nucleus through the nuclear pore, wherein the small and the large subunits now in the cytoplasm and combine with each other and with the messenger RNA during protein synthesis. That means protein synthesis occurs in the cytoplasm of the cell. Another organelle present in the cytoplasm of the cell is the RER or the rough endoplasmic reticulum. And the characteristic of RER, the membranes usually peppered with ribosomes, and because of these ribosomes in its membrane, it is said to be the site of protein synthesis. Another type of endoplasmic reticulum is the soft or the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, wherein the membrane of this structure does not have ribosomes. Thus, the function of this is for lipid synthesis and carbohydrate synthesis, or simple term as lipid production and carbohydrate production. So this is the typical structure of your rough endoplasmic reticulum peppered with ribosomes, and this is your smooth endoplasmic reticulum because in the absence of ribosomes. Another structure is Golgi Apparatus, and Golgi Apparatus are closely packed stacks of membranes that functions to collect, sort, package, and distribute proteins and lipids. Just like in manufacturing plants, there are what we call packaging areas. The same also in our cells. Golgi Apparatus serves as the packaging area where it prepares the proteins, lipids, and carbohydrates that are produced in the endoplasmic reticulum. It simply means that the final touching of the products happens in our Golgi Apparatus. Another, we have the secretory vesicles. And these secretory vesicles are distributed in our cytoplasm, and it functions for the distribution of materials out of the cell. Some materials that are distributed by our secretory vesicles are the products of metabolism of the cell and some other waste products. This illustration is the Golgi Apparatus, composed of flattened membranous sacs and resembles a stack of dinner plates or pancakes. So this one is the transmission electron micrograph of the Golgi Apparatus. Another structure is lysosome. This lysosome functions for the digestion of materials inside the cell. And lysosomes are said to be the suicide bags of the cell because it contains enzymes that digest foreign materials that enters the cell. Also, lysosome contains hydrolytic enzymes that breaks down old parts of the cell. Another, we have mitochondria, which is known as the powerhouse of the cell, and it contains folds, inner folds known as the cristae. And mitochondria is for production of energy of the cell in form of adenosine triphosphate. So, this slide will explain the action of a lysosome. So, a vesicle forms around materials outside the cell through endocytosis. Then the vesicle is pinched off from the cell membrane and becomes a separate vesicle inside the cell. And the lysosome is pinched off the Golgi apparatus and attached or fuses to the vesicle, and the enzymes that the lysosome contains released into the vesicle with foreign material, and this enzyme will degrade or digest and will later on be used up by the cell. This is a typical mitochondrion structure wherein we can observe the cristae or the inner folds containing, of course, the different enzymes needed for ATP production. So this one is a transmission electron micrograph of a mitochondria in longitudinal and cross section. Another structures that are located in the surface of the cell are your cilia and flagella. Cilia, usually many per cell, and it functions for the movement of the cell and at the same time move materials across the cell's surface. Usually, ciliated cells are located in our respiratory system. Another, we have flagella, which has a one flagella per cell, and the function of this is to propel the cell. Like for example, the flagella present in the sperm of organisms. Another, we have the microvilli. Microvilli is shorter than cilia, and the function of microvilli is to increase the surface area of absorption. Particularly, these microvilli are located in the intestinal tract of animals that increase the surface area of absorption of nutrients for the animals. To make the cell a good structure and at the same time with integrity, cytoskeleton is needed. Needed for the cell's framework. And cytoskeleton is made up of proteins. The function mainly to support the cell, usually to hold organelles in place and enable cell to change shape. We have several types of cytoskeleton. Number one, we have microtubules. Microtubules are the largest in diameter among other cytoskeleton, and it provides structural support and usually form the cilia and the flagella of the cell. Another, we have intermediate filaments, and usually medium in diameter, and it maintains the cell shape. Another, we have microfilaments, the smallest diameter among cytoskeleton, and it involves also in cell movement. So these three cytoskeletons work together for the purpose of cell shape and at the same time structures of the cell. I mean, maintaining the structures of the cell. So in this illustration shows the different cytoskeletons, the microfilaments, the intermediate filaments, and microtubules. Notice that these three cytoskeletons or types of cytoskeletons forms a framework that makes our cell organelles intact. After we tackle the basic parts of the cell, let's have a glimpse of the genetic material of the cell. So a cell's characteristics are determined by the type of proteins produced. And the proteins function is determined by genetics, and information in the DNA provides the cell with a code for its cellular processes. And the genetic material of living organism is in a form of deoxyribonucleic acid or the DNA. So the DNA is a double helix which is located inside the nucleus, composed of nucleotides. DNA is a type of a nucleic acid and that contains 5-carbon sugar, which is a deoxyribose sugar, nitrogenous base, and the phosphate group. So this is the typical double-stranded DNA. The flow of genetic information is also called central dogma that occurs in 3 stages: the DNA replication, DNA transcription, and DNA translation. We have the term gene expression. Gene expression wherein the information in the DNA directs protein synthesis or for the production of protein. Proteins provide code for gene expression, and usually enzymes regulate the chemical reactions, and it uses transcription and translation for this. This illustration is an overview of a gene expression wherein the DNA strand here, so the DNA contains the information which is necessary to produce the proteins. And this DNA is transcribed or undergo transcription and may result to an mRNA or the messenger RNA strand, and it will be transported through the nuclear pore going to the cytoplasm and will undergo translation. So in the process of translation in the cytoplasm of the cell, the translation process where the information contained in the mRNA is used to determine the number, kinds, and arrangement of amino acids in the protein. That means it is crucial that the transcription inside the nucleus must produce a correct mRNA to produce a correct protein, which is needed for the development of traits of the living organism. First stage is the transcription. So, transcription is the process by which the DNA is read and usually occurs in the ribosomes. Transcription produces mRNA or the messenger RNA, and this mRNA contains codons. When we say codons, these are set of 3 nucleotide bases that codes for a particular amino acid. We have translation. Translation is a process by which the messenger RNA is converted into an amino acids and forming a long chain of amino acids called polypeptides. And later on, the product will be proteins and codon pairs with anticodons. And when we say anticodons, these are the 3 nucleotide bases carried by the transfer RNA. In short, these anticodons will pair to the codons in the mRNA. This is the standard genetic code that codes for proteins. Notice that we have the 3 stop codons here and 1 codon here that starts the translation. So, the first codon that can trigger the start of translation is the methionine or the AUG. And these 3 stop codons will terminate the translation. In order for the cell to replicate its own, it must undergo cell division. And cell division is the process of forming 2 daughter cells from a single parent cell. Cell division uses mitosis and meiosis. Each cell, except sperm and egg, contains 46 chromosomes, or said to be diploid in number of chromosomes. Meanwhile, of course, sperm and egg contains 23 chromosomes because it will undergo fertilization of these egg cells. One type of cell division is mitosis. Mitosis is a cell division that occurs in all cells except for gametes. It will form 2 identical daughter cells. The components of mitosis are chromatid, centromere, and centrioles. When we say chromatid, these are 2 strands of chromosomes that are genetically identical. And the centromere, where the 2 chromatids are connected, usually in the center. The centrioles are small organelle composed of 9 triplets. So this is a typical centriole. So centriole here is a structure where it is composed of 9 triplets of microtubules. Each triplet contains 1 complete microtubule fused to 2 incomplete microtubules. This one is a transmission electron micrograph of a pair of centrioles, which are commonly or normally located together near the nucleus. So we have stages of mitosis. One is the interphase. In the interphase stage, this is the time between the cell divisions. And the DNA is in the strands, which is in form of chromatin, and the DNA replication occurs. That means in the interphase, there is a preparation of the different contents of the cell before it will undergo cell division. Another, we have the prophase stage. The prophase stage where chromatin condenses into chromosomes and the centrioles move to the opposite ends of the cell. We have the number 3, which is metaphase, wherein there is a alignment of the chromosomes at the center of the cell known as the metaphase plate. Another stage is the anaphase, where chromatids separate to form two sets of chromosomes, and the chromosomes will move towards the opposite side of the cell. Lastly, we have the telophase, where chromosomes disappear and nuclear envelopes and nucleoli will form again, and the cytoplasm will divide into forming two cells. So, in summary, for us to remember the stages of mitosis, we use PMAT. PMAT, that means prophase, metaphase, anaphase, and telophase. And remember that I or interphase is before prophase. So this is the last slide of my presentation, where I am going to discuss the stages of mitosis. So in this stage, the interphase stage, it is the time between the cell divisions, and the DNA is found as thin threads of chromatin in the nucleus of the cell. So the DNA replication occurs during the interphase. Next stage is the prophase stage. So in the prophase stage, the chromatin condenses into chromosomes so that it will be visible already under the microscope. Each chromosome consists of 2 chromatids joining at the centromere. So the centrioles move to the opposite ends of the cell, and the nucleolus and the nuclear envelope starts to disappear. In this stage, the metaphase stage, where the chromosomes aligned in the center of the cell in association with the spindle fibers. The purpose of aligning here at the center is for the balance segregation of the genes for the daughter cells. Next is the anaphase stage. In this stage, the chromatids separate to form 2 sets of identical chromosomes. So the chromosomes associated by this structure, which is known as the spindle fibers, and move towards the opposite poles of the cell, or going to the centrioles at the end of the cell. Lastly is the telophase stage, wherein the chromosome dispensed, the nuclear envelope and the nucleus or the nucleolus is start to form, and the cytoplasm begins to divide to form 2 identical daughter cells. And notice the structure here. If it's animal cell, it is known as the cleavage furrow. If plant cell, it is known as your cell plate. Lastly, in this stage, the mitosis is complete and a new interface will begin. So the chromosomes have formed into a chromatin. And the cell division has produced two identical daughter cells, each with DNA that is the same from the parent cell. So this ends my presentation. Thank you very much for your kind attention. God bless and keep safe, everyone.